All right. Welcome everyone, and let's see everybody here for the December Science Engineering Council meeting. Well, the in-person meetings are working out with the hybrid format. I'm Tim. We've got several board members here today. And we're ready to kick off this discussion. Just want to say first, thanks again for everybody for their renewals. And if you're still interested in joining, as we were just discussing a moment ago, we can assist you with renewals. Either online or by paper, either way. With that, I think we're prepared to go ahead and hand it off to Yvonne to introduce today's speaker and topic and maybe talk about some upcoming events that we have planned.
Great, thanks, Tim. Just wanted to mention that subtitles are available in the Zoom session today. If you choose show captions or transcript or whatever it says in your version of Zoom. You can type questions into the Zoom chat during the talk or ask them after the talk. And we're recording this meeting, and recordings of many of our past meetings are also available on our YouTube channel, so you can check them out there. We'll be letting you know by email soon about the topic and speaker for our January meeting, which will be on January 10th. As always, feel free to contact me about particular topics or speakers you would like to suggest for upcoming meetings. As far as events held by other groups around town, the Las Cumbres Observatory is hosting an Astronomy on Tap tomorrow evening at M Special Brewing in Santa Barbara. And the featured topic is Gaia, 10 years of observing the galaxy in motion. Today, our speaker is Dr. Craig Prater, who is the co-founder and CTO of photothermal spectroscopy. He's been involved in the development and commercialization of novel techniques for micro and nanoscale materials characterization for over 30 years. He was the 2023 recipient of the Williams Wright Award for significant contributions to the field of spectroscopy while working in industry. In 2018, he co-founded Photothermal Spectroscopy Corporation, a Santa Barbara company that has pioneered the field of photothermal infrared spectroscopy. Prior to this, he served as a visionary leader in research, product engineering, and technology development for digital instruments, picometrology, and analysis instruments in the fields of atomic force microscopy and nanoscale IR spectroscopy. Despite being employed in industry for over 30 years, he has co-authored 140 scientific and trade publications and has patents with over 10,000 citations. And he's also been my husband for 34 years. Dr. Prater received his PhD in physics from UCSB in 1992. He is speaking to us today on shining a light on microplastics, providing tools for informed environmental action. Welcome, Craig.
Thank you very much. It's a great pleasure to be speaking again with the Science and Engineering Council. I am really looking forward to discussing this issue of microplastics. It's unfortunately not a very happy topic, but there's a lot of science and technology that is involved in trying to understand and mitigate the problem. So I'm going to tell you, here's sort of the background for my talk. First, I'm going to tell you what are microplastics, what are the origins of microplastics, what are some of the places they come from, and then we'll try to get a sense for the scale of the problem. And you have to think big to understand the scale of this problem. We'll talk a little bit about where we find microplastics, where in the environment, where in the world. And then, why should we be concerned about it? And one of the big reasons that we should be concerned about it is that there are risks to human and animal health associated with the ingestion and breathing of microplastics. And then I'm going to talk about the different ways that the microplastics problem is being studied. With that, I will introduce a local company here, the Photothermal Spectroscopy Corporation, and the instruments that we have pioneered locally that are being used around the world to help understand this microplastic problem. And then at the end, I'll talk a little bit about what can be done to improve the problem, what technologies are emerging that may help reduce this problem in the future. Okay, so first of all, what are microplastics? Microplastics have a pretty simple definition right now. It's any plastic particle that is 5 millimeters or smaller. And so there's bigger names for big chunks of plastics, like a car bumper or old plastic bag, et cetera. But when it gets down into that millimeter, micrometer, and smaller scale, we call them microplastics, and for the smaller ones, even nanoplastics. So where do these microplastics come from? Well, they come from many, many different sources. One of the ones that is most obvious is if you have a large plastic piece of litter, like water bottle, that does not make it into recycling or into the trash, and instead makes it onto the street and washes out into the ocean. The wind and wave action over time will break this, one impact bottle into more and more little pieces. And over time, those fragments get smaller and smaller and smaller. And when they get to the size that they can be ingested by fish or other wildlife or breathed into our lungs, that's when the hazards associated with microplastics start to come into play. Another major source of microplastics is our clothing. So historically, all of our clothing was made from natural fibers. The natural fibers like cotton, et cetera, break down over time and return to the environment. But the clothing that we make from these high-tech fabrics, like our high-tech fleece jackets or high-tech running gear, those are made from polymer fabrics, polymer materials. And over time, in fact, when you wash them, every time you wash them, some of the fibers make their way down the drain into the water. That's actually been a research topic here at UCSB. It's quantifying the extent of that. Also, there's a lot of leakage from plastics production around the world. The sort of the starting material used to make plastic products are called nurdles. They're just little plastic particles that are melted, formed, molded into the products we need. And those get lost a bit in the production process and enter the environment. Historically, there were also personal hygiene products with microbeads that people would use for exfoliation, et cetera. So these have been more recently phased out because of the understanding of the environmental hazards associated with. And what's the problem with all of these is that a lot of plastics and other industrial chemicals are called forever chemicals. The issue is that they just don't break down over time. They break down into smaller and smaller pieces. They become more and more hazardous, but they don't decompose the way that like a natural fabric or paper material would. And then another really big source of microplastic particles is from tire wear particles. So basically what happens is we're driving down the road, that every 50,000 miles, you need to get yourself a new set of tires, and that's because you've worn the tread off of the tire. Well, where did that tread go? That tread broke down into little tiny particles that got deposited on the road, and then when it rains, those wash down into the creeks and watershed that make it out into the ocean, and then get accumulated in seafood or in sea life, et cetera, and then can make their way into our seafood. So microplastics, starting roughly about 10 years ago, the awareness of microplastics really started to take off, and now there are thousands of news articles every year on microplastics. There are some that I've pulled out from recent ones where there have been concerns about, all right, how much microplastics are there in our takeout food that's washed into our rivers and oceans, et cetera? How much is in the the food that we eat? How much is in the ocean? And recently, even finding it in the human body, in blood, for example. And this is some work from, this was actually from about 10 years ago, here in Santa Barbara, looking at microplastic pollutants in the seawater. Actually not in the seawater, but in the offshore sediment. So basically what they do is they dig down into the sediment, they filter out the sand and see what non-sand particles exist, and they can find a bunch of microplastics particles. And they notice these, like you go to the earlier sediments, they don't find microplastics, but from World War II and beyond, when we had a big uptick in the production of plastics, they start to find these microplastics in the soils. Okay, how big is the problem? I saw this graphic the other day, and this is fairly horrifying. This is a representation of the total amount of plastic waste that is expected to be generated between 2010 and 2050. And to give you a sense of scale, down here on the bottom left, this is supposed to be Manhattan, One World Trade Center there. And this is how big the plastic wastes are that we are producing. Okay, well, you think, well, we're making all of these plastics. It's not that big of a problem, because every week I fill up my recycle bin, I wheel it out to the curb, and it's taken off, and I pat myself on the back and I feel good. So here's a question, a quiz for you. What percentage of plastics do you think are being recycled right now?
2%.
We're putting all of this into our bins. We've got to be close to 100%, right? No, this guess is good. You're a little bit more pessimistic than the actual number. But right now, only about 9% of the plastics that we put into the recycle bin get recycled. And so about half of it is dumped in landfills where it will stay forever. About 20% is incinerated, which produces pollution, toxic chemicals, and even microplastic particles for the unincinerated particles. And then about a fifth of the plastics that we produce as waste is doesn't make it into the sanitary system, so it ends up on beaches and roads, et cetera. Okay, and then why is this? And in the U.S. it used to be about 10% in that last graphic that I showed you, but more recently it's down around, I think, 5%. I can't see the. Okay, and the reason is that China used to be the big market for acquisition of our plastic waste. They had a whole bunch of recycling plants there. They put a moratorium on the import of plastic waste. And so suddenly our market for plastic went away. And so what that resulted in is about a cut in half of the total percentage of plastic recycling in the US right now. Now, there are attempts to build up US recycling plants, but that really hasn't come online yet. So right now, about 95% of the waste that we create is not recycled, and about a fifth of it worldwide ends up as litter that turns into microplastics. It's interesting to compare that, though, to paper. We are much more successful recycling paper. So the plot on the left here is the production of paper, and then the percentage of paper waste that's recycled, which is about 75%, whereas it's in the 5 to 10% for plastics. And so if we need to get plastics to the same recycling level as paper, or go to alternate materials that don't last forever like the plastics do. Okay, so this is what the flow of plastics are typically. So we start out with fossil fuels, almost 100%, 99.5% of plastics come from fossil fuels, from oil. And then you can see the different trajectories where about 50% of that is goes to the landfill, about 20% is incinerated, About a fifth of it is mismanaged, as I said, goes into to litter, and then historically, this is an older chart. Well, about 50% collected for recycling, but only about 6% are actually recycled and turned into new products. Okay, so that means that what we're worried about primarily is this mismanaged waste that's going into our waterways and air, et cetera. And to give you a sense of scale of the 2.5 billion metric tons of solid plastic that's produced around the world, that ends up about 8 million metric tons of plastic going into the ocean every year. And let's give you a sense of scale of that again. 8 billion. Between these two, so I don't know which is right, but the scale they're talking about, over 150,000 times the weight of the Sydney Harbor Bridge, or a billion elephants, et cetera. So very, very large number. Another way to visualize that is equivalent to dumping one garbage truck into the ocean every minute. And another horrific statistic that I saw is that this is equivalent to lining up 5 trash bags every linear foot of coastline around the entire world. So that's how much plastics we are putting into the ocean every year. Okay, so where are these microplastics found? Bad news is pretty much everywhere we look. So this is a plot from of all of the locations that have been sampled so far looking for microplastics. And the reddish orange dots are places where microplastics were found, and the yellow dots are places that microplastics were not found. So you can see there's hardly any yellow dots. So basically almost everywhere we look, anywhere in the world, we are finding microplastics. And where are we finding it specifically? We are finding it in water. We are finding it in streams and estuaries, and lakes and oceans. We can sample it from the air, so we are all breathing some level of microplastics. It is accumulating in the soil around the world, and it's found on some of the most remote places on Earth. So even in fresh snow in the Antarctic, we are now finding microplastics. So this really is a global environmental problem that our use of plastics has created. So now I'm going to talk a little bit about how these microplastics are sampled, measured, analyzed, et cetera. And so typically what happens is you have an environmental researcher that goes out into the field in one of these locations, either on a boat or hiking or in an expedition to Antarctica. They dig up samples, they bring them back to the labs, and they filter them and then examine them for the content of microplastics. So this is an image of some microplastic particles in snow in the, I don't know if this is in the Antarctic or this may be from the Alps, but you can see a variety of different fibers and colors of fragments of the plastics that have made it into the snow. We are also finding microplastics in many foods and beverages. Unfortunately, bottled water is one of the worst, as I'll show you in a bit. But we're also seeing it in other beverages, in table salt, which makes sense if you think about we have microplastics in seawater. We evaporate seawater to make table salt. So the plastics tend to get concentrated in the table salt. We see it in a lot of seafood because a lot of seafood, they mistake these little plastic particles for food particles. So they eat them and then it makes their way into their organs. And because we have microplastics in the soil, we can also have it in the foods that we grow there, especially the root vegetables have been known to accumulate microplastics particles. And then because we have microplastics in the air, those microplastics particles can land on flowers, and then when bees are out collecting pollen, they collect microplastic particles in their with the pollen that then makes it into their honey. And so we have detected microplastics in all of these foods and beverages that I've indicated. To give you a sense of scale, you know, we drink bottled water oftentimes because we picture that we're getting this, you know, pure spring water product. But the problem is that, you know, water is spending a lot of time in a plastic container, and those plastic containers are shedding into the water in addition to any microplastics that were already in there. So you can see, for example, in this 2019 study, they found about 100 grams per liter or meter cubed of microplastics particle in bottled water versus only 4 in tap water. So you're actually, if you're concerned about microplastics, you're actually healthier drinking water out of the tap than, you know, putting it into a bottle like this with no plastics in it. Okay, and then we have also been finding microplastics directly in the human body. There have been 2 studies recently, one looking at blood samples, another looking at breast milk, and in about 3/4 of the subjects in both of those studies, microplastics were found in the blood and the breast milk. So we're breathing these things, and then they're going into our body and unfortunately going into our babies as well. One of the, I'm going to come back to this in a bit, but one of the things that people study a lot is the distribution or the concentration of particles versus size. And so they typically will make a histogram like this, where on the x-axis is the size of the microplastic particle, and then on the y-axis is the abundance of each size, or the, you know, the concentration. And typically these curves have a pseudo bell-shaped curve where there's some peak, and so you would naively look at this and say, okay, well, most of the microplastics are between 100 and 150 microns, and so we just need to worry about those. Well, the problem with this plot is that what this actual concentration of microplastics looks like is this. It just keeps going up and up and up. The smaller the particle, the more particles there are. And the problem is that there is a detection limit of the instruments that we use to measure microplastics. And when they get too small, we can't detect them anymore. And so we think, oh, there's no plastics there. But it turns out there's way more small plastic particles than there are big. And the reason is that basically it's just a simple counting thing. If you take a macroscopic piece of plastic and then you break it into 2 particles, now you have 2 particles. Now, if that breaks down further into smaller particles, now you have 4. It breaks down smaller, now you have 16, et cetera. So the more times these little microplastic or these plastic particles are broken up by sun and, you know, ocean waves and things like that, the more plastic particles they make, and then the smaller they get, the more dangerous they are, because they can penetrate further into the human and animal bodies. I'll talk a little bit about that in a minute. Okay, so how are we exposed to microplastics? And it's pretty much every way that we can interact with the world. So through the inhalation of contaminated air, through drinking and eating contaminated beverages and food. Really small particles can make it through the skin. And then how this becomes more dangerous is, you know, normally we have a variety of barriers in our body that prevent particles from getting into the bloodstream, et cetera. And so if it's a big plastic particle, it would just pass right through your body. But if it's a small enough plastic particle, it will pass through the gut barrier, and that can make it into the bloodstream. And if it's a small enough particle, it can make it through the blood-brain barrier and into the brain. And then there's the same thing with the blood-air barrier in your lungs. And then there's open questions about, you know, can it pass through the placental barrier and impact children in the womb as well. So there are a very, very large number of studies underway that have been completed trying to quantify the risks to humans and animals associated with ingestion and inhalation of microplastics particles. And we unfortunately don't know the answers right now. So I can't tell you what what your risk is of getting cancer because of exposure to microplastics or lung disease, et cetera. These are not quantified yet, but a lot of people are working on it. The things that we do know is that the average human is inhaling about 160,000 particles a year and ingesting about 50,000 particles. But that is based on the particle sizes that we can quantify now. The actual number is probably much, much higher because, again, we have trouble quantifying these smaller particles. Okay, so what are we doing about it? Fortunately, we live in a proactive state, and California back in 2018 passed the California Senate Bill 1422. And this requires, so it recognized that there's a risk associated with microplastic particles and required quantifying the microplastics that exist in our drinking water. So basically, they're first of all trying to establish what are the safe guidelines for the amount of microplastics in the water, and then doing testing of all of our public water sources to make sure that we are below those thresholds, and then if we're not, trying to find out ways to mitigate and improve, et cetera. So this has led to a lot of the research that my company and other companies are doing to help provide tools for being able to analyze the microplastics in the environment. Okay, so what I've already touched on some of this, but what's the scope of research that's being done regarding microplastics? So first is where are we finding microplastics? What's the concentration? And then there's a lot of interest in the distribution of the different plastic and rubber types to get to the next question, which is what are the origins of these plastic particles? So if you find like a big concentration of plastics in some area, can you track it back to the source so that you can figure out how to cut off that source, or replace the plastic that's causing the microplastic particles with some environmentally friendlier material? And then what are the environmental impacts and what are the health impacts of these microplastics? And then finally, once we understand the scope of the problem, what can we do to make it better? UCSB has been an active area in microplastics research. And I have some pictures here of some of the folks that are doing microplastics research. Actually, their names have disappeared. But as I mentioned, one of the things that they have been working on, this is Patricia Holden, and she and colleagues worked with Patagonia, looking at this microfiber issue of the synthetic materials that end up in our, washing out the drain of our laundry and into the ocean. And so they were working with Patagonia to try to understand which materials are the worst shedders, what can be done to reduce that environmental impact of those performance materials. And then this gentleman here, Mark Anthony Brown, he was a postdoctoral researcher back here at UCSB, and he was looking at the ingestion of microplastic particles by little sea critters and seeing how they bioaccumulate. As I said, because these small sea animals and even large fish are mistaking microplastics particles for food and then incorporating them in their bodies. Okay, so how is some of this research done? It's done, as I said, by environmental scientists around the world. So if you're looking at microplastics in the water, there is typically some kind of collector like this that somebody on a ship is going to throw overboard and drag behind for some amount of time. And there'll be a mesh collecting net that the water pass through, but then collects the microplastic particles. These types of things, though, again, you tend to undercount substantially the smaller particles because the smaller particles are just going to pass through the holes in the net. So they're not a great way of sampling the potentially riskiest particles. I mentioned that there's a lot of interest in where in the world do we find microplastics. So this is a plot. The short answer is that while we find microplastics pretty much everywhere we look, the highest concentrations are near urban areas, as you would expect. That's where we're generating most of the waste and where it's mismanaged. So this is around Vancouver Island, BC, and this is showing an example plot of the concentration of microplastic particles. So these numbers here are these numbers are the locations, but the colors here are the concentrations. So you can see the orange area close to the city, and the inlet to the city has the highest, and when you get further afield, you get down to lower. So it's pretty clear that the origin of the problem is our cities, but the impacts are felt for thousands of miles away. There's also, as I touched on earlier, lots of interest in microplastics in soil. They will look at soil in farmland, under estuaries and riverbeds and beaches, etc. And so typically one will collect, almost like an archaeologist in some regular way, and then filter down the sand and analyze it. So the process looks sort of like this. You have your raw sample, which you will then filter out so that you filter out the larger particles, and then you have some smaller particles left. It turns out that a lot of those particles are not microplastics, because if you go out to the ocean, there's lots of small particles that are not pollution. They're pieces of sand, broken down seashell, little bits of decaying plant matter, plant and animal matter, etc. And so then there are techniques that people use, typically by density separation, to try to get the microplastics to float up to the top, leaving the dirt and heavier material behind. And then eventually you pass the resulting water through a filter, and then stick that filter into a microscope to analyze the distribution of the different microplastics particles. And so a typical report looks something like this. There will typically be a series of optical micrographs or optical microscope photographs that show the different types of shapes and colors of particles. And so you'll see fibers and fragments, etc. And then there'll be an analysis by size, and then oftentimes an analysis by polymer type. And this is the information that can give us hints about the origin of the material. For example, this, where is it? PET, which I'm not seeing on this list, is the type of plastic we use for plastic drinking bottles, and nylons and polyesters and acrylics. It could be used in clothing, etc. Rubber is going to be in tires, etc. And the color can also give us potentially more information about the potential use, whether it's a packaging, etc. And so, coming back to the topic or the title of my talk, shining a light on microplastics. So a very typical scenario is that you have scientists looking under an optical microscope, and they are, trying to identify and quantify the different microplastics. And so, these are the types of things that we see: fibers, which would be from clothing; film, which would be from, for example, like food packaging; And fragments would be bigger pieces of plastic that got broken down over time. And then these micro beads, which were for the exfoliation products. Now, unfortunately, and so in the early days, the analysis was typically done by optical microscope, and the scientists would look at that and say, well, that looks like it could be polyethylene, and maybe that one is polystyrene, but they didn't know for sure. And it turned out that they made a lot of mistakes in that sort of eyeball analysis. And so there became a strong interest in doing quantitative chemical analysis so that you could definitively know what the different polymers were. And so one of the techniques that was used is called pyro GCMS. And this is, so pyro means heat, and GC is gas chromatography, and MS is mass spectroscopy. But the basic idea is that you would heat these plastics till they broke down into their constituent molecules, suck them into a vacuum system, and use strong magnets to bend their ions as a function of the mass, and then you can get a series of peaks that will give you a indicator of what the chemical composition is. The problem with this technique is that it is very slow and it's completely destructive because it vaporizes the material for you to analyze it. And so this is not a good technique for analyzing large populations of microplastics. If you have ones or twos, it's okay, but not if you're trying to analyze thousands. And so that brings me to the technology that my colleagues and I have developed. We developed a product called the Mirage, which is using an optical microscope-based technique to do spectroscopic analysis of even some of the smallest microplastic particles. And I'll talk about how that works in just a minute. So a little bit of background. Our company, as many companies in Santa Barbara, take advantage of infrared light. And specifically, we use a technique called infrared spectroscopy. So starting with the fact that light is a wave, it's an electromagnetic wave that basically is oscillating up and down with a given frequency. It turns out that if you take that light in the infrared wavelengths and you impinge it on organic molecule, it turns out that the wiggles in that light wave correspond to natural frequencies that the molecules will bend and stretch at. So it's just like these are things called resonant frequencies. For example, if you take a slinky and you bounce it up and down, there's like a natural frequency it wants to bounce at. Well, there's many orders of magnitude faster, but there are frequencies that molecules like to wiggle at. And so if you hit these molecules with light at the right frequency, you will cause it to oscillate. And so if you measure the amount of light absorbed by a particular sample as a function of the frequency of the light that you're wiggling at it, you'll get a series of peaks that correspond to different molecular bonds. So for example, the molecular bond of nitrogen and hydrogen has a specific frequency it oscillates at. The same for carbon and hydrogen, and whether they're single bonds or double bonds. And so for any given material that you analyze with infrared spectroscopy, you get a series of peaks that acts like a fingerprint that you can use to uniquely identify a specific material. What we can do is we can take these are called spectra, infrared absorption spectra. We can take these and put them into a computer materials library and say, what is this material? It'll tell us what the best matches are. And then we can use that to identify these different plastics. And there's, that all sounds great. There's one problem, and the problem is this thing called the diffraction limit. And the diffraction limit says that if you are trying to focus your light down to the smallest spot that you can so that you can measure this tiny little microplastic, there is a minimum diameter that you can focus the light to that is based on the wavelength of the light. And this kind of grumpy looking guy, Ernst Abbe, was the first person to write this formula down, and he got so famous for it that this is on his memorial. Basically says that if you have you can't make measurements on things that are much smaller than the wavelength. And unfortunately, infrared wavelengths are relatively long. They're big compared to the size of lots of microplastics. And so if you take a sample with some fine structure and you put it into a regular optical microscope using visible light, like we can all see, we can see down to very, very fine features. If you do the same measurement in the infrared, you can see how these fine lines blur out. And that's because of this optical diffraction limit, that we just can't see really small things with big wavelengths. Okay, so Santa Barbara tech company to the rescue. We locally here in town commercialize a technique called optical photothermal infrared. And so it works in the following way. We use a microscope and we focus infrared light down onto the sample, and we can't focus it as tight as we might like. We can focus it only to about 10 microns, which is about 1/10 of the diameter of a human hair. But then what we do is we come along with what's called a probe beam. We use green light, which has a wavelength that's about 10 times or 20 times shorter than the infrared light. And so we can focus it 10 to 20 times smaller. And what happens is when we tune this infrared beam to that vibration frequency that excites the molecular vibrations, we will get heating in the sample, and that heating in the sample can be detected with this visible probe beam, now with much finer resolution on a much finer scale than we could with the infrared. So this technique lets us go down to particles that are even 0.5 micron or smaller. So that gives us the ability to analyze plastic particles that we couldn't with other techniques. And then coming back to this curve here, we can now measure these particles that were in this undetectable region right here, the smaller particles. So we can give much more accurate assessments of the distribution of the different microplastics. Okay, so we're based in downtown Santa Barbara on Chapala Street. We have this cute little building with a recently replaced Spanish tile roof. I work in a lab where we build first benchtop prototypes. So this is kind of like a tinker toy setup where I order things from an optical supply company and put them together like tinker toys to demonstrate the capabilities, new technology. And then once we have something working well, our engineering team will turn it into a nice looking product here. And we've been very fortunate to have been pioneers in this area and have a lot of patents associated with the underlying technology in partnership with various universities and national labs. So this OPTIR approach has been was introduced commercially in 2018, and we've been really excited by the number of scientific publications that have been coming out every year using this technique. This is through 2022, but I think 2023 were something like 150 publications per year so far. And the scientists using this instrument have had their research featured on the covers of a bunch of prestigious scientific journals. And then this OPTIR, this infrared photothermal technique, has been used in a wide variety of applications, a lot of them in the life science. I'll touch on a couple of those, but microplastics is one of our biggest application areas. So just a couple of other applications before I talk about how this is used for microplastics. We've been doing a lot of biomedical research with this instrument, and this is showing some measurements related to neurodegenerative diseases. And so it turns out that in the patient, a patient who has a neurodegenerative disease like Alzheimer's or Parkinson's, et cetera, there are plaques in their brain tissue that are composed of protein aggregates or misfolded proteins. And we've been able to use fluorescent dyes that highlight where those are, and then use this spectroscopic technique to analyze the chemical composition of these plaques and compare. This is called the conformation, basically how these proteins are folded, and we can compare normal tissue and diseased tissue to quantify the different effects. Our instrument has also been very heavily used in cultural heritage, including looking at Van Gogh paintings to understand the details of the pigments and how different artworks are aging over time. What are the impacts, et cetera. And then on the industrial side, we have a lot of Fortune 500 companies that are buying our instruments because they are producing materials that are very sensitive to defects. If there's a defect, it'll make the product not work right, like your computer chips or your flat panel displays, et cetera. So we have a lot of those companies that when they have something that's not working right, they go and find the defect and analyze what it is. They take spectra, compare it against the library, and they can say, aha, this is a polyethylene particle. This must be coming from this step in the process. We've got to go clean up that process. And then also a lot of interest in the forensics and security front. Okay. So back to microplastics. So we've seen a very large number of publications from our research community using this OPTIR technique to look at microplastics. And I'm just going to touch on one of them that got a lot of press for reasons that I'm sure people will understand. There was a group in China that analyzed the baby bottle nipples, which, the common thing that one does if you're sterilizing them before giving your baby milk, you put them in a pan of water, boil it, and now you've sterilized it. Well, what these researchers found was that process generated microplastics that then the babies could be ingesting. And so they were able to test that process and quantify the amount of microplastics and the type of microplastic particles that were coming off from that steam disinfection. Okay, so we have recently added a lot of automation to the microscopes to make it so that scientists can efficiently analyze a large population of microplastics. So typically how this works is that they'll filter the microplastics down onto a filter, put it under the microscope, and then use one or more optical technique to basically see where the microplastics are. And then they can choose which plastic particles they want to analyze based on their size and shape. And then the microscope will automatically go to each one of these particles sequentially, take an infrared absorption spectrum. So these are the infrared absorption spectra, and you can see how these profiles have different shapes depending on whether the material is a PMMA, a polystyrene, polypropylene, polyethylene, nylon, polyimide, et cetera. They all have very characteristic fingerprints that we can discriminate. And then in the end, we generate a report which says, okay, here's how big the particle was and what the material type was, and make a report for them. And we have done that for even local environmental samples. I went off to Goleta Pier, not far from here, lowered a bucket into the water, filtered some of it down, analyzed the distribution of microplastics, and found things like high density polyethylene, PTFE, which is Teflon, epoxy, And then, one of the complications I mentioned earlier is there's lots of things that are not plastics, like sand and bone and shell and things like that. Okay, in the last few minutes, I want to touch on, well, what can we do about it? So it would be nice to think that we could just go out into the ocean, scoop up the microplastics, put them back in a landfill, and pat ourselves in the back. And there are people that are working on things like this 3D printed microplastic filtering robot. So they have a little robot that swims around the pond, sucking in, filtering out plastics. Unfortunately, this type of approach, as cute as it is missing a key thing that we call scalability. If you were to try to make enough plastic fish robots to cover the world's oceans and have them try to eat little plastic particles and not affect the environment in some other way, that's unfortunately a pipe dream, I think. There are ways to potentially have nature help us out. So a lot of people are studying, are there ways to engineer bacteria to help break down plastics. So there are researchers that have, it's kind of a funny story. They found a garbage pile on their university campus that had some plastic bottles or something that had been there for years and years. And so they went and they took soil samples and cultured the bacteria that they found in there. And they found some bacteria in there that over time had evolved to eat some of the plastics that it had nearby as food. Bacteria turn out to be exquisite at evolving themselves to take advantage of their environment. So through selective culturing of these bacteria, people have developed bacterial populations that can be used to break down plastics. And so there's ongoing research on that. There are also looking at the materials that the bacteria are using to break it down, which are certain enzymes. Can you just use those enzymes? So there's a lot of research on using enzymes as molecular scissors. Basically, polymers, plastics are chains of carbons and hydrogens in complex configurations. And if you could have molecular scissors that would just go and flip the bonds between the carbons and the carbons and the carbons and the hydrogens, eventually you just get back to carbons and hydrogens, which would be non-toxic. And so there are people who are studying ways to break down plastics into their constituent parts to be non-harmful. There are also people looking at plastic eating enzymes for what's called depolymerization. So what I said is that plastics and polymers are these long chains of carbons and hydrogens and other things. They are chains of identical units. So when you have something, for example, it's called a polypropylene. That means poly means many, and propylene is the subunit. So you have lots of these propylene monomers. And so one of the barriers to efficient recycling is that it costs too much money to take our waste plastics, break them down into a form that can be used for new plastics. And so right now it's cheaper to use new oil and a lot of energy to make new plastics rather than the breaking down the old plastics. But that's just a cost calculation. And so various researchers are working on ways to efficiently, with low energy, break down specific plastics into their monomers, into their building block units that then can be reused to make new plastics efficiently. And then there are also people working on these photocatalysts. So photocatalysts are little particles that typically use interaction with sunlight to initiate a chemical reaction. And so people are working on these catalytic microparticles that when you have microplastic in contact with it and you have the sun, then it could help break it down. And then one of the efforts that I'm most excited about is the possibility of high performance biodegradable plastics. And so there's this gentleman, Sao Noda, that I just had the pleasure of speaking with at a conference. He, when he was at DuPont, developed this material that's called Nodax. And it basically uses bacteria that grow plastic precursors within their bodies, so it can be done pretty efficiently. But then these materials, while they'll be stable over the shelf life that you need for a consumer product, when they get exposed to sun or water or compost pile, they break down very, very quickly. And so there's this company, Danimer Scientific, that has been founded to commercialize this Nodax plastic, and they already have a plant online and are looking at scaling up to much, much larger production. And it's already being adopted by some big brands. So if you want to help reduce microplastics, you can go drink some Bacardi rum and do your part. Okay, so with that, I'd like to conclude. So first up, microplastics, I hope I made it clear that it's a global environmental problem that has profound impacts in the environment and potentially unknown risks to human and animal health. There is research going on around the globe to try to understand the distribution of the different microplastic particles, where they come from. And then there is lots of exciting and enabling research here in Santa Barbara and UCSB, our company, and other areas that are looking at the characterization of microplastics. And then, finally, we all need, as technologists and consumers, to innovate, to come up with new strategies to mitigate the problem and make things better for the future. With that, I'd like to thank you for your attention.
So, questions?
Yes.
Questions in the room or in the Zoom?
First of all, extremely informative, just outstanding plus. The presentation is just marvelous. I learned a lot.
Thank you very much.
And I want to thank the chair lady for finding me. And my comment is, it's just spinning in my head, but I think mathematical modeling and simulation could be a real idea for having parallel efforts, because you're collecting data which allows some validation of the model and simulation as it is evolving. Your thoughts?
Yeah, absolutely. So approaching the microplastics problem is definitely a multidisciplinary approach. And so at the conferences that I go to where microplastics are being discussed, you have physicists, engineers, environmental scientists, biologists, mathematicians, people who are doing modeling and simulation. So absolutely all of these approaches are being integrated to try to address the problem.
I'd like to talk to you further after the session about that. Thank you.
So what would you say the percentage of these microplastics that are bioinert, like TMNA is basically bioinert? So, and any sense for how many particles are versus are not?
Yeah, so the question is about what percentage of these materials are bioinert, and that brings up a big issue that I forgot to touch on. One of the big hazards associated with microplastic particles is even in the cases of bioinert plastics, when you put them in the environment, when you put small plastics in the environment, they tend to accumulate the toxins around them. And so there's a lot of research that's going on to looking at microplastic particles in the environment and seeing, other forever chemicals like PFAS that are attaching to the surfaces of these microplastic particles and then getting ingested by seafood and increasing the risk. It's not my area of expertise, so I couldn't give you a quantitative breakdown, but basically the plastics that we find in the environment are very similar to the distribution of plastics that we produce. So the ones that we produce the most of, for example, polyethylene is very heavily found in the environment. PET very heavily found in the environment. Tire rubber very heavily found in the environment. Yes.
I was wondering, in your chart, you had showed that you got like another order of magnitude smaller in size. So I imagine you want to measure about 2 orders of magnitude, maybe 3 smaller micro nanoparticle sizes. So what's the pathway to measuring the smaller?
Yeah, so previously, my colleagues and I had a company in town called Anisys Instruments, and that company made a photothermal infrared microscope that could go a couple of orders of magnitude smaller using a technique called atomic force microscopy. And so you can do the same approach of illuminating the sample with infrared light and then using the probe of an atomic force microscope to measure much smaller particles. And so that is the approach that will most likely be used for the smallest, nanoparticles. But there's, right now there's still a tremendous amount of interest in the micro from, millimeter scale to, 1 micron or so, which is a large population of plastics we're trying to understand.
Mary, you have a question?
Yeah. First of all, I'd like to say it's very clever, Craig. I'm impressed with the idea of using visible light or fluorescence to concentrate your material and then be able to use IR. Very clever indeed. And then I've got a personal question. Personal question is there any difference in microplastic concentration in bottled beer versus canned beer?
Okay, so there are 2 main sources of microplastics in beverages. The first is how much is coming in the water, and then the
Right.
second is how much comes in the
Container.
the container. And so I'm sure people are doing that study. I don't know the answer, but I can comment on a couple of things. So that
I would presume that the amount of microplastics in beer would have in part to do with where the brewery is located and what kind of water is being used.
Well, don't
Yeah, well, so
cans
one would-
have plastic linings?
Yeah, exactly. Cans have a
Yeah, I
plastic lining.
I was gonna get to that. So, your starting point is, tap water,
Exactly.
which is cleaner than, the end products. And so, one would think that if you had glass bottled beer, it would not be much worse than the tap water, except if there are microplastics being introduced by the hops, for example.
Exactly.
In plastic bottled water, you have the packaging, and as other people just mentioned, in canned beer, there is a plastic liner on the inside. I am not personally aware of studies yet of an analysis of whether that is leaching into the beverage, but I'm sure that that's underway if it hasn't already been studied.
Okay. Thanks, Craig.
What about biodegradable plastic, like the vegetable bags that you get? Is that really beneficial?
So it depends on the type of the biodegradable material and what environment it ends up in. So some of the biodegradable plastics are biodegradable only if they're in a aerobic composting environment. So if it has enough heat and water and air or whatever to let it break down. And so a lot of the materials that go to a landfill and get, buried where they don't have access to water or it's anaerobic, those materials don't break down very quickly. That's one of the things that this company, Danimer's product, is advertising, is that they are able to break down in lots of different environments, including in a landfill, in water, in sunlight, et cetera. Just unfortunately, they're not yet scaled. You can't go to the store now and buy bottled water in this material, but hopefully in the future, both them and their competitors will make, truly biodegradable products available, more widely available.
Another simple question. What about cellophane? Cellophane used to be very popular and polyethylene has replaced it. But was cellophane good for the environment?
That's a good question. I don't remember enough about cellophane, but that brings up a question. So why do we have so many plastics? The reason that we have so many plastics is because they are cheap, strong, temperature stable. They can be made flexible. They can be made rigid. They can be made shock absorbing. So what's that? Clear, colored, painted, printed on, et cetera. And so it's for all of those, highly desirable properties that they have replaced, metals and wood and cotton fabrics, et cetera. So while I don't know the specifics of cellophane, my guess is that the performance that we could get with, modern engineered plastics was better than we could with cellophane.
Especially the cost. And the last question I had is, what's the average, if that's the right word, cost of such instruments?
Oh, the microscopes are in the hundreds of thousands of dollars, a few $100,000, depending on the range of capabilities.
Yeah, it would seem like the technology used to make the microchips, the lithography, like they're working with very small things. You can see, and then with ultraviolet light. It seemed like that. Have you tried any of that sort of thing? Is there anything that would get you orders of magnitude more resolution, wouldn't it?
Ultraviolet light has a wavelength that is not that much shorter than visible light, and so it would give us a slight increase in spatial resolution, but infrared light also has a lot more ionizing energy in it, and so it tends to destroys the sample. Yeah, I mean, it might not be an issue for the plastic samples, but a lot of our customer base is in the life sciences, and they don't like things being irradiated with ultraviolet light.
Yeah.
George, you had a question about the
Yes.
biologic.
The question, what do we know about the biologic effects of microplastics and what harm they cause to biology?
Yeah, so that's an extremely active area of research, and the details are a bit uncertain. But the things that we know is that when the particles are inhaled or ingested, they can make it into the bloodstream. They can go past the gut barrier. They can accumulate in organs. This graphic here shows some examples of recent publications that are looking at accumulation of particles in lungs and digestive system and et cetera. And so the thing that we don't know yet is what increase in disease or mortality or et cetera is associated specifically with microplastics. And the problem is that humans are exposed to a million environmental factors of how much time did you spend in the sun, how close were you to a copper smelting plant, what was your nutrition when you were a child, et cetera. So it's really hard for epidemiologists to control for all of these factors and say, okay, your microplastic risk is this. And so what they're tending to do right now is do more things like animal studies, where you have simple animals like a a clam or, some small sea animal. And then they will intentionally expose them to known quantities of microplastics. And then they'll start to measure at what concentration, was there an impact on their reproduction or their growth or their lifespan, et cetera. So those are the types of experiments that are being done right now to try to quantify at the simpler level and give us insights for what's more specifically to look at for human health.
So is that something that UCSB Marine Sciences is doing?
UCSB Marine Sciences is absolutely sampling microplastics in the environment and trying to understand, what's in the water, what's in, various sea life, et cetera. In fact, they were over at some of the UCSB researchers were over at our facility for a demonstration and some measurements on their samples recently. Yeah, Jim.
Greg, first of all, thank you for letting us know that beer is 3 times safer than bottled water. I'm going to use that as an excuse to drink more beer than bottled water. But also, I found it interesting you're talking about the microplastics in the bloodstream. And is there any evidence that the microplastics have bound with other substances that might be in the bloodstream that would people would have more strokes, for example, because the particles are combining together and then constricting the flow of blood?
Yeah, I mean, that is absolutely an area of active research. I don't know the details, but that's one of the big questions is if you get microplastic inside an organism, how is it interacting with the body? And yes, are things, binding to it, or is the microplastic binding to something that's blocking some, critical biological function? Those are the types of things that people are doing a lot of research on.
Is there a number for the amount of particulates released by tires?
There is, but it's very location dependent. But I have, for example, some measurements that were done in Germany. If I can get to them. Okay, yeah, here's an example. So this is specific to Germany, but they did a study of microplastics and where they thought most of the particles were coming from. And in this particular study, they thought that 88% of the particles they detected were from tire particles. I've seen other studies where it's more like a third. But yeah, it's the tire.
We don't have like the 4 million tons of tires are in the air. We don't have that kind of a number.
Oh, I'm sure you could get to that kind of number. I mean, you could do basically if you figured out, the lifetime of every tire and how much tread wore off the tire and how many particles that broke down to, that all goes into the air, into the water. None of that makes it into the landfill.
I bring it up because one of our recent speakers was an expert on oak trees, and one of his problems was finding a breed of oak tree that would stand up to tire particulates.
Oh, really?
And still be able to blossom.
Yeah, interesting.
Any other questions from the Zoom?
Hi, Craig, an excellent presentation. How much of the microplastic problem or issue could be solved by taking care of the macroplastic issue? And, you showed your chart of particle size versus number of particles. It might be helpful to have a chart of particle size versus mass. There are places in the world that still just dump their trash in the ocean, dump their trash in the river. And it would seem like that sort of thing would be the first low-hanging fruit that we could use to eliminate not just the microplastic problem, but obviously the macroplastic problem.
Yeah, so it's absolutely true. I don't know if I have it in here or not, but yeah. So
There you go.
here is a map of the mismanaged waste by region. And so you can see that, China and Southeast Asia, so the darkest colors means a greater percentage of mismanaged plastics that go into the environment instead of being recycled or into landfills. And so, yes, absolutely, by controlling things at the source, we can certainly have a big impact. And, China, to their credit, they're one of the most active research communities worldwide on microplastics in the environment. And so I have no doubt that we will see, this number, go down over time. You're absolutely right. The more that we do one of two things, either reduce the amount of plastics that we are manufacturing, and two, reduce the amount of plastics that are being mismanaged and entering the environment, we will make the situation better.
Thank you.
Yes.
If I understand what you're saying correctly, it seems like the question is, no matter how fine your analysis gets down to biodegradable plastic, it sounds like it has an impact on future plastics, but does it really impact current microplastics?
Yes, that's an extremely good point. So, if you ask what can be done about the microplastics that are in the ocean now, unfortunately, nobody that I know of has any really good ideas. Because you have these tiny little particles that are globally dispersed, and there are, an abundance of other particles of similar size that naturally appear in the environment. How do you suck up the plastics without sucking up the food that the sea life is eating? So it's a really hard problem. And so for some of that, we're going to just have to wait for those things to, break down to the point that they're small enough that they're less of a hazard. But the projections are, with the rate at which plastic production, plastic waste is increasing, we're going to be contributing to, doubling the problem in the next 50 years or something like that. And so, we can, the things that we do now will prevent it from getting much, much worse in the future.